Okay, so second part, dedicated to cell therapy. So the first person is Patrick Hainaut, co-founder of MSL.
C'est toi.
Hello, can you hear me?
Il y a un petit décalage.
First slide.
Hello,
Are you connected?
I'm connected and I can see you and hear you. Tu m'entends? Okay.
Yes.
First, we would like to thank the committee of organization to give us the possibility to introduce the project of MSL, which involves the development of a therapeutic tool composed of NK cells amplified and activated in vitro. I must precise that this introduction is a short introduction with 3 slides. On the first slide, at the top of the left, you have the 2 mechanisms of action of NK cells to lead the lysis of the target cells. You have this the natural cytotoxicity with the balance of activator and inhibitory signals, and you have the ADCC mechanism with monoclonal antibodies coated on the target cells and by CD16 on NK cells. Close to this diagram, we give an overview of the clinical trial using NK cells according the origin of the NK cells. In fact, we observed that 77% of the clinical trials are using NK cell, mainly obtained from HLA selected donors and very often relatives. On the middle of the slide, at the left, we plug the number of starting clinical trials according the time. This is just to show that there is an increased interest in NK cell therapy. In the beginning of the 2000 years, we find only 15 clinical trials using NK cells, and in the last 4 years, we found more than 100 clinical trials using NK cells. So it seems that the interest in NK therapy is increasing. What we try to develop in MSL is a product with the following specificity: low toxicities to treat fragile patients. We want to develop a generic allogenic product. We don't need donor selection and manufacturing for each patient, and specific manufacturing for each patient. And we want to have optimized efficacy. And for the first part of the development, we don't want to genetically modify the cells. What we try to do, in fact, is to develop a product, a therapeutic tool, with to be used in association with monoclonal antibodies at the beginning, and with other molecules later. Regarding the source of NK cells, we use cells from umbilical cord blood. And the reason why we use the NK cells from umbilical cord blood are as following: availability of the source, safety of the source, low GVHD risks, and low viral transmission risk, and no patient-related HLA selection of donors needed. Next slide, please. Okay. Concerning the project, we can present the following figures with the different steps. First, we use NK cells due to the poor toxicity of the cells. We use allogenic NK cells, and we also use expanded activated alloreactive NK cells, and we can optimize and modify the quantity of the quality of the cells. These cells and these NK cells are derived from a pool of umbilical cord blood, and we got delivered patent on this process, and this patent is delivered in Europe, actually in Europe and Japan, and not yet in United States. One of characteristics of the cells, according to the process we have developed, is the quantity of CD16 expressed on the NK cells, which allow ADCC. And we got a product with two components, as we want to use these NK cells in synergy with the monoclonal antibody. So we got a product with our NK cells and monoclonal antibodies. Regarding the clinical development, we plan to clinical trial by using NK cells plus rituximab to treat CD20B lymphoma. And in the second part, we plan to use these NK cells plus cetuximab to treat colorectal cancer. Next slide, please. Okay, just a very short statement regarding our development. We have split into two parts. The product development step and the POC steps. Regarding the product development step, which is a very, how can I say, very difficult part for the NK cells, we have performed a process at lab scale. We have performed the industrialization of this process, and we contract the production of 2 mid-scale batches, which allow us to realize proof of concept on different monoclonals, with different monoclonals for different tumoral pathology, and also we have to subcontract the full-scale batches productions, GMP upgrade, and clinical production. Regarding the proof of concept, we are working on different subjects. First, regarding the B lymphoma, we perform the in vitro concept in vitro and in vivo. And we have to perform a clinical trial phase 1 and 2. And we have to move forward for the development for colorectal cancer. This development plan has been discussed in scientific advice with the French regulatory agency. We also work in the lab on multiple myeloma, and with also trastuzumab to treat gastric cancer. but it's just the beginning of the development. In fact, when we consider the NK cells, you can use this NK cell used with classical monoclonal antibodies that we call anti-PA, or you can use multi-specific antibodies and immunocheckpoint inhibitors. We can try to improve the NK persistence and activity of the NK cells by two systems. The preloading, in fact, the coating of NK cells with different molecules, maybe fragment of MAB or maybe NK activator, or we can perform genetic modification, for example, to increase interleukin 15 expressed on the membrane of the cells. This is a global overview of our project. Thank you very much.
Thank you. Now we have I'm checking my note. Yes. So Jean-Antoine Ribet from Bluebird Bio from States. We need to thank you because he had to wake up very soon in the morning. It is yes, it is 4:30 for him. So it's a very good example because it's a company connected to Europe, based in the States, and still very, very involved in Europe, very, very successful. And I have a pleasure to introduce him and to ask him to speak about his company, Bluebird.
Okay, thank you. Can you hear me well? So can you hear me? Yes, I think so. I've got the echo. Okay. So first, thanks to the organizers for allowing me to present remotely. I've got the echo. Can you cut the echo, please? And be sure that I would have loved being with you in Paris. It's really here, really early in Boston. So I will focus my presentation on what I've been doing every day for almost 2 decades, which is gene therapy for sickle cell disease. As I work for Bluebird Bio, I have some disclosures. So please consider this when I present the results of our current studies. So please, next slide. Yes, you present the slide. So this is my disclosures. So can you go, please, next slide? Yeah, thank you. I chose to work in this field because I realized that hemoimmunopathies are a growing public health problem all over the world. Sickle cell disease is one of the most monogenetic blood disorders in the world. The incidence of sickle cell disease is higher in the southern hemisphere. It is estimated that each year more than 300,000 babies are born with sickle cell disease. The distribution of patients in Europe varies greatly from country to country, and France and the UK are the most affected. The US is also greatly affected with over 100,000 patients nationwide. Please, next slide. Sickle cell disease is an autosomic recessive gene disorder caused by a specific mutation in the beta-globin gene. This mutation produces a specific strain of hemoglobin called hemoglobin S. Normal hemoglobin is always soluble in the red cells, but hemoglobin S is characterized by polymerization under stressful conditions, such as low oxygen levels. Such conditions induce red blood cell sickling. Sickle cells do not flow normally through blood vessels because of their decreased malleability and stickiness caused by their abnormal sickle shape. They can cause blockages in blood flow, which in turn leads to painful vaso-occlusive crises and hemolysis. Vaso-occlusive crises and hemolysis affect the whole body, leading to severe chronic complications, organ failures, and significantly shorter life expectancy. Next slide, please. Up to now, the only curative treatment for hemopathies has been hematopoietic stem cell allogenic transplant with a matched sibling donor. But most patients don't have access to allotransplant because they don't have a compatible sibling donor. And these procedures are associated with high risk of morbidity and mortality related to the transplant by itself. This is why gene therapy strategies that use patients' own modified stem cells have been developed. Several strategies are currently in development. Gene addition, which is the insertion in the DNA of gene sequence to produce a functional protein and restore normal cell properties. This is what we develop at Bluebird Bio for sickle cell disease, thalassemia, and adrenoleukodystrophia. Gene editing that modifies the DNA to correct a mutation or induce other mutations to restore normal cell properties. And gene regulation, which leads to target modifiers of disease severity. Next slide, please. You can see by this timeline that there has been a collective effort these last 30 years on this major goal to cure hemopathies through gene therapy. First, Yves Bezard did a postdoc in Cambridge, Massachusetts, in the MIT lab of Vernon Ingram. Vernon Ingram was one of the pioneers of molecular biology and had determined the hemoglobin S molecular structure. Then Yves Bezard returned to France to work on mouse models for hemoglobinopathies. One of his students, Philippe Lebouche, went to Harvard University in Cambridge to design the lentiglobin vector, and he proved the concept by curing thalassemia and later sickle cell disease. After that, the first protocol was set up with the first patient treated in 2007 in Paris. And finally, Bluebird Bio worked on improving the process and designing new protocols, the first of which was conducted at Necker Hospital in Paris. Next slide, please. The BB305 lentiglobin vector we use is a lentiviral vector, which is a combination of 3 HIV genes, a CMV promoter, and the envelope of another virus. The lentiglobin vector carries the human beta-globin gene. This gene sequence has been modified with a specific mutation to produce hemoglobin A-T87Q to induce anti-hemoglobin S polymerization properties. The patient's hematopoietic stem cells are put in contact ex vivo with the lentiglobin vector, which generates the cells and then incorporates the human beta-globin gene into the patient's DNA. Next slide, please. This flow chart summarizes our gene therapy process. First, we harvest hematopoietic stem cells. We collect peripheral hematopoietic stem cells by apheresis after mobilization using Prexafor. I need to clarify that this use of Prexafor is off-label. Then we select CD34 cells and transduce them with the BB305 lentiglobin vector. We test the quality of this drug product while these transduced cells are frozen. After that, the patient is hospitalized and receives busulfan chemotherapy to achieve myeloablation. Finally, the drug product is infused intravenously and the patient is hospitalized until engraftment has been achieved. Then we closely monitor each patient for a period of 2 years and conduct a long-term follow-up study for a period of 3 or 13 years. Next slide, please. The HGB-205 phase 1/2 study was conducted in Paris. It was the first study that treated sickle cell or thalassaemic adult and pediatric patients with the BB305 lentiglobin vector. In this protocol, everything was centralized in the same building at Necker Hospital. By that I mean the bone marrow stem cell harvest, the cell transduction, the cell infusion, and the patient's follow-up. 7 patients were treated at Necker, 4 patients affected by a beta thalassemia major, and 3 sickle cell disease patients. Next slide, please. After the results for the first patient treated at Necker with sickle cell disease, Bluebird Bio set up a multicenter phase 1/2 study in the US called HGB-206 on adult patients with symptomatic sickle cell disease. I'll show you the results on the next slides. Following this, you can keep this slide, no worries. So, following this, a confirmatory phase 3 study called HGB-210 has been launched in Europe, the UK, and the US on pediatric and adult patients. I won't overwhelm you with tons of scientific results, but I will show you the most significant ones. This shows the percentage of red blood cells that contain the specific immunoglobulin 87Q circulating in the patients after a gene therapy transplant. These cells are represented by this mix of blue and orange, representing a mix of immunoglobulin 87Q and a mix of immunoglobulin S in the same cell. You can see that after 6 months, more than 70% of the red blood cells circulating contain immunoglobulin 87Q, and after 18 months, this level reaches 90%. We consider that even 60% is a positive result. The immunoglobulin S and immunoglobulin 87Q distribution in these cells is comparable with cells of asymptomatic carriers, AS patients. Next slide, please. And this is one of my favorite slides, which is called the swim lane. Each swim lane represents a HGB-206 group C patient transplanted with a lentiglobin vector, BB305. In blue is the pre-transplant period. In yellow, the post-transplant period, and the diamonds represent a severe vaso-occlusive crisis or acute chest syndrome, which is a severe complication of sickle cell disease. You can see that there haven't been any acute chest syndrome or severe vaso-occlusive crisis in any of the patients since they got transplants. One patient had one non-serious grade 2 vaso-occlusive crisis 3 months post-treatment, but has had no relapse since then. Next slide, please. The safety of the procedure was comparable with an autologous transplant with no major concerns. The only adverse events were related to chemotherapy. There was no adverse event related to gene therapy. The insertion site analysis showed polyclonal hematopoietic reconstitution without clonal dominance, and no lentivirus replication was detected. Unfortunately, 20 months post-transplant, one patient with a history of severe sickle cell disease and severe pulmonary hypertension died suddenly. This death was not related to gene therapy, but was apparently caused by pre-existing cardiopulmonary problems. Next slide, please. This example with sickle cell disease shows one of the ways we at Bluebird Bio work to recode science and medicine to improve the lives of patients with severe genetic diseases, such as sickle cell disease, thalassemia, and adrenoleukodystrophy, and for those with cancer. Thank you for your attention.
Thank you. So now we have the round table. So I'll ask to the different speakers to come to the booth, and also the two chairpersons. Jean-Charles Soria had an obligation. You know, he was very sorry to don't come, and he asked to Patrice to replace him. Patrice is a member of the new management committee, you know, at Gustave Roussy. So I think he's the right person to replace Jean-Charles. So we have. So we are missing somebody, no? Yes, we have. Who presented? No, I think we have. So all other people are connected, as many other people from around the world connected. I don't know how many, because large majority of attendees are connected, you know, as you know. So for this roundtable, perhaps I shall start by asking if the room has some questions, you know, because we had no questions between for the specific speakers. So if we have questions, perhaps now to feed the roundtable subjects. So I have myself many questions, but perhaps we could start by asking to you if you have questions. The first one is the more difficult. Alain, you are the person to ask the first question. No question? Okay, so I shall start with my questions. You know, I have many, so I shall try to limit myself, because after, you know, the concluding remarks will be made by Patrice, you know. So I was very impressed by because you know, perhaps I have with those technologies since many years. I contributed to the start of Selectis, for example. I've been chairman of the company during many years. I faced the problems which have been listed concerning manufacturing, price, and all those things, you know. So I have many questions. But first, 30 clinical trials in phase 3. Frédéric said that it was mainly for rare diseases and cancer, oncology. So a question I have to the specialist: do you think that those technologies, which are now very developed more than 20 years ago or 40 years ago, do you think that those technologies could be applicable to any diseases? You know, you spoke about it, you know. So I was very impressed, because it's true, if you look at the current application and the drugs in clinical development, they are relatively limited to rare disease and cancer, even if there are many other applications coming, and we saw it for CNS, for example. So could you answer to this question? Because, you know, when I was young and partly managing Sanofi, you know, we were with small molecules, and only a small part of what we were developing were large molecules, or gene therapy or cell therapy was not even existing. So it has changed a lot, you know, when you look to what is proposed for approval, you know, to the FDA. So could you answer to the question? Marina, you want to answer?
Yes, it is a very good question, even if I am a great optimist. So the question to your question is, we can cure any disease because the creativity of the human beings is pretty enormous. So I think as long as human beings are creative, we have a solution for any disease. The importance is to know very well the physiopathology of the disease, the medical issue that we try to solve, and to put together competence from a different field, immunology, virology, technology, algorithm, and so on, to make the process successful.
Thank you.
May I add something?
Yeah.
Yes, I think, yes, it's gene therapy, as I said in my talk, is not only for rare genetic disease, it's just medicine, it's just like small molecule. It just has very practical opportunities. One is to be able to go specifically in one cell type or in one organ, ultimately, of course, if we make a lot of progress in that direction, but it will be the case. The second thing is it can be modified as will, deliver in the long term first, but also in the future with modification that will be made, stop production when you want or induce delivery when you want. So it's really an extremely powerful tool, but it's not different from classical medicine and from small molecule. And the second comment is that you have to also to know and to think that gene therapy is not only one gene, one protein, and that ultimately it could be combined approach to combine pathways that could be synergistically targeted to treat a disease. Because medicine is, for severe disease, medicine is never one drug for one disease, like in cancer, for example, or infectious disease. So gene therapy will be the same, combine synergistic target to treat diseases, severe diseases.
And what is really impressing me with the long story I have in the pharma industry, you know, it's that the success is coming from research, the development. You know, I have no doubt that it will be resolved. But after that, you have a long process with many other expertises, you know, it's involved. You know. Technology, platform, manufacturing, purification, market access. After you have other funding, you know, we have many large venture capital funds, you know, connected or attending, you know, and it's large amount of money, more than ever, you know. And after that, you have the pricing, reimbursement, access to the population. So you have many. It's very complex, and it will need very intelligent people, including at the political level, you know. So it's very complex. Patrice, you want to?
Yeah, because
it's working?
yes. Can you hear me?
Yeah.
So because there is something we did not discuss. You just discussed about all the stakeholders in the chain, but there is something we did not discuss is regulatory agencies. And for me, it looks totally incredible that when you do gene replacement therapy, you have to go to regulatory agency for each replacement, right, of gene. But I think on the long term, what is going to happen? Because you cannot make a drug for each very rare disease while you are using the same vector and the same strategy, in fact. So are you going to go through something that is more global, where you say, we regulate the process instead of each gene that is replaced?
No, that's a very good point, and it's a critical one. In particular, when we're talking about rare or ultra-rare disease, I mean, the agencies and the overall mindset in terms of regulation is really inherited from small molecules and large indications, meaning that the way you present now a regulatory file for gene therapy has a very in-depth, for instance, CMC, so the product, the way you manufacture the product. I mean, this is something important, the way you design your clinical trial. All the principles are really inherited from large indications and small molecules. And for rare, and it comes to a point that for some indications where you would have only a handful of patients to be treated in Europe every year, the cost of bringing the drug through a registration is such that it can be detrimental for patient access. So you have here a clear issue, and Nathalie alluded to that. I mean, we are developing these drugs for genetic disease with a certain number of patients, spinal muscular atrophy, Duchenne muscular dystrophy. We're developing them for frequent disorders such as cancer. But now back to rare and ultra-rare disease, if we have to find a regulatory pathway that allows access without putting hurdles that are just considered too costly to get the products to the patients. So that's clearly a challenge.
And by extension, and it's a question for Dr. Moti. You mentioned that you can fine-tune what you can do in CAR T-cell by knocking out some gene, adding some gene. But it's the same question. What is the regulatory path for this? Because ultimately you can change 10 or 15 genes per CAR T-cell, but how are you going to move forward in terms of regulation?
I know this is really a very important and very good question. Obviously, when it comes to the regulatory, I think
Please speak louder, because we have difficulties. So I don't know, is it technical people could improve it, but we are not. Start again.
Okay. Yeah. No, I think this is really an important question because, as I mentioned, we can. I'm speaking very, very loudly. I can't do more.
It's okay now. It's okay now.
Okay. No, I think it's an important question because, as I mentioned, the number of combinations is an endless number. However, from a regulatory perspective, I think they are treated like drugs, like any molecule, let's say. So the regulatory agencies, and that can be maybe a pity, but we have to live with it, would consider, in my opinion, every variation as a sort of a new drug. Although I'm not an expert in regulatory approval, or I can't speak for the regulatory bodies, but for the moment, although, for instance, from a clinical standpoint, we do not see major differences between products in a given indication or a given disease. Still, these products are considered totally different drugs with their own development and so on. So, and I think multiple myeloma that I mentioned is a very good example for this, and that is using a lot of resources because people are targeting the same antigen, the same disease setting. They will get probably the same label, the same wording, but we have different competing products, and these products are managed and handled as different drugs. So that adds another level of, I would say, complexity, wasting of time sometimes, and of course, resources, consuming resources.
Thank you. Any other comments? No? Yes? Another one?
I think that it is important for the patient to know that their safety is highly protected. So I am not sure that I am in agreement with the fact to sort out cell or gene therapy from the standard track to have a drug approved. Because this is a very well done legislation in order to allow the patient to be protected enough from any not founded innovation, not so well-founded innovation. So say that we can skip steps for very rare disease is not a good way to proceed for a safety issues and for the equity of access to improvement for any patient.
Thank you.
Let me just
comment
on that. And of course, I mean, I think no one should compromise on safety. This is clear. But just let me give you a specific example. According to GMP regulation, when you do a batch of a product that is to be injected, there's a part of the samples that have to be secured for future QC. In the case of gene therapy products, when you do a gene therapy batch, one batch will serve you to treat 1, 2, or 3 patients. I'm talking about viruses. And if you follow by the book the regulations, half of the batch should be secured in some instances for QC. So it clearly was not the intent of the regulator to have half of the batch, because when the regulator thought about keeping samples for QC, it was for large batches, for small molecules. Here, if we agree with the fact that we have to put on the side half of a €1 million batch only for QC, and QC that might never happen on the long term, this has an impact on cost. So of course, it's not about, adapting regulation is not about compromising with safety, but just thinking about what exactly these products are and how the regulations should adapt to these products. And if not, they're just, I mean, the cost, the financial hurdle to get drugs for ultra-rare disease will be such that either there will be no drugs or they will be priced at prices that will not be affordable.
Marina?
I think it should be keep attention to claim this type of observation, because we are discussing success of gene therapy. But if you look at very precisely into each clinical trial, there is also a lot of failure. And failure can be because gene therapy doesn't work, because the dosage of used for the patient is not adapted, even because the AAV vector injected intravenously was so high that the patient died. This happened also recently with the Audentes clinical trials. So it is absolutely necessary to go step by step toward the authorization to put a drug product into the market. And for all these reasons, it is necessary that the authorities and the patient can go back to the drug product to really understand what's happening to the patient. So I think probably there is something in the middle, as often in the medical application of the innovation, where safety and the possibility to go back to what has been injected back to the patient can be reanalyzed at the light of a side effect. And not saying there is a few patients, so we can conserve or cryopreserve or maintain very small budget for eventually analyze the clinical drawbacks. And I would like that the gene therapy field should be much more open to disclaim also failure and the difficulties, drawbacks, and bottlenecks, because to avoid to distribute the idea to the patient and population that need this innovation, that everything is going very well and we cure every patient we have treated by gene therapy. So caution has to be stay our key word in this complicated field.
Thank you. I am relatively optimistic with regulatory bodies, attitude, because I think one good consequence from this epidemic, is the fact that they became more flexible and adaptable. Having developed vaccines and other things in my life, I can say to you that what I have how they manage, this regulatory work in front of companies for vaccines, for example, in US, for example, the FDA, it's very, very new, never seen before. So I hope that for rare disease with people to treat urgently, I think that they have started to, I would say, to change their mind. And also the pharma companies also, because when I was with Sanofi, and I know that other companies, they were relatively strict in terms of, now they are more adaptable, they are collaborating more, changing, and I think it will help, to have products approved faster and perhaps in a less theoretical approach. Okay, so second question is concerning manufacturing. As you know, manufacturing is a big challenge, for those technologies. Only one number, I discussed recently with different large investors, because many funds, American funds, European funds, Asian funds, Israeli funds, are looking for this sector, the CDMO sector, to manufacture for gene therapy and cell therapy. And the challenge is huge, because you have the capacities available today, to manufacture those products, are only 1/3 of what is needed, by the different companies, the large companies. And when you look to what has happened to large and talented companies as Novartis or the things, during the last years, I can say to you, I faced it, at Selectis when I was a chairman, and it's really a big challenge. Manufacturing, yields, quantities, quality, reproducibility, cost, all these things are very, very challenging. And I saw the name of Universal, I know very well, because since many years the founders are close to us, and in France we have Geneton, we have Flash Therapeutics, we have in UK we have Oxford, so we have companies. So how do you foresee that? Because again, it will be not successful if we are not able to manufacture it in quantities. And as it was said, the quantities are huge, and the doses are the concentration are very, very different from what was existing with the vaccine industry. And the costs are still very, very high, and reproducibility is still a problem, I know less in gene therapy, but in cell therapy, I have faced it, So it has consequences. So how do you see that? What has to be done? Because if you don't collaborate yourself with a technology company to develop new system to manufacture, I don't see how it will be successful at the end. The market access will be very difficult since the beginning. After you have other problems, one treatment, several treatment, price, reimbursement, So perhaps.
Yeah. Yes. Can you hear me? Yes. Okay. So yeah, the manufacturing aspect is it's usual to say that the supply chain is important, but here in the case of gene therapy, we have seen in the past month, you could say, dramatic consequences linked to the difficulty of biomanufacturing. For some of the drugs, I will not go into names, but for some of the drugs that have been approved recently, they have not been given to patients just because the sponsor did not have the appropriate capacity, or because the sponsor did have problems in manufacturing. And we have seen, from an ethical perspective, we have seen very bizarre situations where the sponsor did organize lotteries to choose who amongst the patients could benefit from the treatment. And this was just because the drug was not available in sufficient quantities. And I think nobody, of course, us at Geneton being funded by a patient organization is something that we've been very shocked about and that we've been communicating, but I think nobody wants ever to be in that situation. So it means, as you said, not only increasing the capacity for production, but it means also coming with novel technology, disruptive technologies. And here, the difficulty is that it's not only the question for industrial affairs. In large pharmaceutical companies, manufacturing is a question for industrial affairs. Here it is about innovation which is at the crossroads of virology, cell biology, equipment, artificial intelligence, sensors for analytics. So it's really a trans-domain work and innovation that has to be done, that has to be organized. And that has not only ethical value, as just mentioned, but also economical value, because whoever will have a patent position on these novel methods for production will also be in a very strong economical situation. And I mean, whoever, I mean, both from a company perspective, but also from a country and geographical perspective, in sense of independence and autonomy for production, being able in France and in Europe to master this production capacity is critical for our pharmaceutical
It's very,
future.
very critical. Marina?
Unluckily, there is not only the limitation you say about the problem of choice the patient to be treated. There is a lot of other bottlenecks. For example, provide North America competitor with exclusive license of a product developed in France. This is another bottleneck that make our patient not to be treated by innovation developed here. Other limitations are not a regulatory agency, but how the industrial company have organized the flux of a patient, because now if there is a gene therapy treatment available in North America, we have to make the patient flight from Europe to North America with all the constraints linked to pandemic infection. This is unethical. This is really unethical to make a patient in very bad clinical situation, to oblige him to fly in another countries to have a treatment that could be provided here. The example as a physician, I can give you a lot, name and also companies, profitable or not profitable, that have put some bottleneck to treat the patient. So we have to be cautious when we give example, because example can be multiplied at the infinitive to show where is the block. This is one of the reasons, I think, much more than fight against regulatory authorities, that I think they do their work to protect the patient. We have to work to avoid the shipping of a patient. Patient doesn't have to put in a flight to go to have a treatment in the other part of the world. This is the reason that our company, Smart Immune, make a special effort to develop technology that permit the patient to be treated in their country and in their own hospital. This is the future for the patient and for the access. And this machine that can automatize the treatment can also have a huge impact on the pricing of the gene therapy and the cell therapy, because the procedures invented from very talented engineers can use a very small quantity of antivirus, of cytokines, to reduce the price of thousands of euros per patient. This is a durable development that we have to sustain.
Thank you.
I think this is true for all gene therapy products, actually, not only for what you just described, but it's true also for production of AAV vectors, for example. And the manufacturing innovation will be shown in the future years. And for example, as I showed, storing the products in room temperature, or storing the product on a very small device that allow you to make it travel around the world. This is also something currently done, and that will improve a lot. And Marina is right, the patient don't have to fly to the product, but the product has to be given to the patient locally, and this is exactly, I think, what everybody has in mind right now.
Okay, I have to stop the roundtable to let to Patrice the time to conclude. I had many other questions concerning market access, business model, you know, but I think it will be for the next event. You know, we are planning to organize another one next June. You know, so I hope it will be possible. So it will. Nevertheless, the subject we are discussing today will still. Perhaps be partly resolved, but still existing, and with your project. So thank you for that. I'm sorry, but it's too short. But you know, we have another meeting after, and we need to let the time to come here to conclude. And so the difficult task to conclude those difficult subjects.
Well, I'm not going to be very good. So first, thanks everyone for having attended the meeting, and thanks for the speaker. I'm not an expert in gene therapy.
It's why you are here.
Yeah, but I learned a lot of things. I am a medical oncologist seeing breast cancer patients. So you see, I'm very far from rare disease. So I'm sorry if sometimes I use these logics of oncologists. But I've seen several points that I would like to highlight. The first one, and I'm sorry to come back on this, Marina, but it's a regulatory question. I understand regulation is here to protect patients, but is also here to accelerate drug access and time from development to access. It seems to me there is something to work at the regulatory agency level. And I agree with you, the most important is to preserve the safety. At the same time, it seems to me that all this question of gene replacement therapy, we could maybe broader each disease, you know, you know, if we add many very rare diseases, we can come with something more frequent, we can have the same regulatory pathway. I think there is some work that can be done around this question of accelerating development. The second question was about the scale-up, and here, again, I fully agree. The drug should come to patient. I was very shocked by hearing that some patients have to travel to get drug access. So upscale, it's still unclear for me what is a general bottleneck, but I'm not sure it's technology. It seems to me it's more investment than technology, but I don't know, maybe you can make a comment.
No, I think it is a technology
Okay.
overall.
So the
investment in all the technology that can just increase the price is not a good investment.
Okay. So point number 3, yes?
Just comment.
Yeah.
Because, I mean, we're here at the European conference, and I think we have to consider that, I mean, Europe is still lagging in terms of investment as compared to the US, where the investments are huge for gene and cell therapy, I mean, counting to billions. But here, there's still a lag. I mean, still hope, but a lag.
Point number 3 is access. I mean, I work in a field where only few patients in the world have access to innovative drug, and it seems to me it's a little bit the same situation here. So I think when we further develop the drug, we have to keep in mind that 95% in the world do not access our drug, and this is a major issue we are facing now. Most of beautiful science you are doing will never reach most of the patient, and I think this is we need here a workforce, I mean, a lobbying for this problem. It's a huge problem, you know, and it's not only US, Europe, you know, it's a huge problem. The fourth point that I'm not sure I'm going to be able to verbalize it is the question of incremental change. We have the discussion with CAR T-cell. I don't think that we will be able to go one gene per gene. I think it's time to really dramatically change the CAR T-cell and maybe go to a package of 5 to 10 changes in gene and manufacture this.
Perhaps a comment?
Yes, of course.
You know, it is changing.
You are the moderator.
Also,
so
because it is I have been always interested by the third world, by the developing world. I can say to you that venture capitalists are thinking about it, and I know some of them. There are many Americans, but also Israeli and Europeans also. They are more and more structuring their funds by having a for-profit fund and a charity fund, you know. and using a part of the profit to fund the charity funds. Gates did it with a fund which has just invested in Universal, you know, and I know some other projects. So I think that even, you know, the capitalist world, you know, is thinking about it because he knows that he is not addressing that. Nevertheless, the problem will come to us, you know. So it's very important,
yeah.
And changing, you know.
Yeah. So the point about incremental change that I was mentioning for CAR T-cell is very simple. The way we are developing drug now is phase 3 randomized trial, and it's absolutely impossible that modifying one gene is going to improve outcome as compared to a control. So either we modify a full package or there is no improvement. Point number 5, only have 2 points after, don't worry. It's drug versus cell therapy, and that was a question for Sebastian, because it's still unclear for me why he decided to move the path of cell therapy while he had an obvious molecular target for a drug. So I think this question of where we move in terms of development is a question. I had a question about gene replacement and immune response, but I think you discussed a little bit this point. And the last point I wanted to make, I was very impressed by the capacity of your two centers. You know, I mean, Imagine and ICM for developing startup from your own science. And I think this is a model that should inspire a lot of other centers in France. And I know my center is trying to do the same. It's not yet there, but I think it's a source of inspiring for most of the big centers.
This is very, very impressive. You know, as I said, I started my first company 2 weeks after my PhD, you know, but it was in 1974, you know, and I was perhaps the second one in France, and I became a venture capitalist after. And I must say that it's very new, you know, even when I was director at Pasteur, you know, I was obliged to push the people, the scientists, to start companies. Now it seems to become normal, and it is, for me, it is perhaps the best news, you know, I could have, because nevertheless, the large companies I know very well also are less innovative, and to have that sort of startup starting and collaborating after, or making any sort of, and you have now available funds coming more and more, so I'm very pleased for that, because, you know, at the end, what is important is to manage, you know, to have drugs on the market to cure, you know, diseases which are not cured. You know? So it's very important. So I'm very pleased to have scientists more and more involved in companies' development. You know?
Okay, and my last point was really the tagline. I love the last slide. That was record for life. I think it was, the general conclusion of the session. So thank you for your attention and enjoy the conference.